Right, I think we'll kick things off now. Normally we wait till about 2 minutes past, and we've reached that time now. So let's go ahead. So thanks to Dr. Rob Noble for joining us here today. So this is the Tissue Image Analytics Centre seminar series, and we always invite a speaker to give a presentation along the lines of something relating to computational pathology, which of course leads us to our great presenter today. So just to say a bit about Rob and his background. So he first obtained his degree in mathematics, where he then earned his PhD in mathematical biology at the University of Oxford, investigating interrelationships between host population immunity and pathogen population genetics. For Rob's first postdoctoral position with Michael Hochberg at the Institut des Sciences de l'Evolution de Montpellier, he switched his primary focus from infectious disease to cancer. He chose to pursue a second postdoc in Nico Bierenwinkel's group at ETH Zurich within a systems biology department to unite dynamical models of somatic evolution with methods of molecular data analysis and phylogenetic inference. Rob later took a concurrent postdoc appointment in Hannah Koko's group at the University of Zurich using life history models to understand the evolution of cancer risk as part of the International Arizona Cancer Evolution Center. Then since July 2020, he's been a lecturer in applied maths at City University London, where he's continuing his research at the intersection of mathematical, computational, and theoretical biology. So with that, it's our great pleasure to welcome you here today, Rob, and give a presentation. And yeah, whenever you're ready, please go ahead.
Thanks very much. I didn't realize that, yeah, my product biography on my website is maybe a little wordy. I might edit it down a bit, but thank you very much for going through that. So welcome everybody. Yeah, so my background, as per the introduction, I started off in mathematics and I've moved into cancer evolution. I see myself as a bit of an evolutionary biologist, computational biologist, theoretical biologist, and a mathematician. I do a bit of each. And the talk I'm going to give you today touches on each of those specialties. So I now just have to share my screen with you. There we go. So do you get a full screen of the slides?
Yeah, looks great.
Excellent. Okay. So the topic is explaining modes of tumor evolution. And the main problem that started this work is thinking about tumors as an evolutionary system, which is at the core of everything I do. There was a debate that has been going on for several years now about what is the mode of tumor evolution. That means what is the broad pattern in this evolutionary system. So several modes have been proposed, and there are various degrees of evidence for these different modes in different tumor types. And these parallel the modes of evolution that were previously discussed in organismal evolution over the 20th century. The first idea is that tumor evolution might follow a punctuated pattern, whereby some big events happen early on, and then there's more or less stasis as the tumor expands. So the evolutionary trajectory of the tumor is set very early, and its composition is set very early. The second case is branching evolution, whereby the evolutionary tree has a few branches, and these persist for quite long periods of time. The third is linear evolution. So in this case, what happens is new variants emerge, new clones, which are fitter than the previous clones, and these achieve selective sweeps. So they replace most or all of their predecessors. So we get a succession of selective sweeps. And finally, a fourth possibility is neutral evolution, whereby the mutations that occur as the tumor grows, after the tumor has got going, it doesn't acquire any mutations that spread more than others. So the fate of a mutation largely depends on when it emerges and not its fitness. And there has been a debate, as I say, about which of these predominates in various tumor types. The greatest evidence is for linear evolution very early on, for branching evolution in the majority of solid tumors, and for neutral evolution, perhaps in some cases. The extent to which branching evolution versus neutral evolution occurs is still debated. Punctuated evolution can be seen as a transition from linear evolution to neutral evolution, and I'm not going to talk about that case too much. But what you see here is just a cartoon. And at the time I started this project, which is around the time this review was published in 2017, there was no particular mathematical or theoretical model which explained why we would expect one of these patterns to come to predominate instead of another. So that's what I set about doing, was creating a computational model into which we could put the basic properties of tumor evolution and then see what kind of mode we get out and how sensitive the mode of tumor evolution is to the properties, the evolutionary or ecological properties of the tumor. So this is the basic workflow. We start off with some assumptions about the spatial structure, the cell dispersal type, and evolutionary parameters. Those structures and parameters go into an evolutionary system that's either my computational model or that's a real tumor. That generates an evolutionary history. We then summarize that, because usually for tumors, we cannot access all the information about the tumor's entire evolutionary history. So we want to just have some summary statistics that are useful. And we do the same thing for the computational model so that we can compare. What we then get are some summary indices, and if we're lucky, a phylogenetic tree. And then we use that summary information to categorize the tumor's evolution, and from that categorization, we get an evolutionary mode. And this system allows us to compare the results of a computational model with real clinical data, as we'll see. So what I created was a flexible model of tumor evolution. This is an agent-based model. It's stochastic, so you can run it many times with the same parameters, and each instance will be different. And the most important thing is it has a flexible spatial structure, which means I can use this same framework. Yep, we got something. Yeah, bias. Okay, so I've got a question here about a sweep. What is a sweep? Yeah, so a selective sweep happens when a new mutation occurs, and because it confers higher fitness, it replaces its ancestor, the previous type that lacked that mutation. So for example, the omega variant of coronavirus has achieved a selective sweep more or less by replacing delta, and the delta variant before it replaced the previous variants. That is a selective sweep. So if you look at a diagram of the frequency of each of these types over time, what you see is that delta took over. It started very small, and then it gradually reached close to 100% in the UK, and then omega did the same. Each of those events is called a selective sweep. And the same thing can potentially happen in tumors, that new clones arise and they replace everything that was there before.
Great, thanks, Rob.
No problem. So as I say, the strength of this model is that within the same framework, I can recapitulate different spatial structures that we see in tumors. So this allows me to just parameterize this model with a small parameter file that has the evolutionary parameters in it, and then some parameters which control the spatial structure. And that allows me to fairly compare these different models all within the same framework. Because the issue is that people had previously modeled tumor evolution, but the standard was to assume one particular spatial structure, various underlying model assumptions, and then to vary some of the parameters, but not to vary the structure of the model itself. And what was new here is that I could change the model as well as changing its parameters. Okay, I see something else in the chat. Sweep, an evolutionary coup. Yeah, that's another way of seeing it. Yeah, it's taking over. Something else I can do with this model is I can look at tissue invasion. So many models of tumor growth, what they do is they start with cells that just expand into nothingness. And they assume that the parameters are such that this is a reasonable representation of the tumor invading tissue. What I did instead was I started out with a domain that is entirely filled with normal tissue, and then you introduce a transformed cell to the middle, and that cell actually has to compete with this normal tissue. So you explicitly model tumor invasion, tissue invasion, rather than making it implicit. Yep, there's a hand up.
Yeah, sorry, just one more question from somebody in the room. So they want to know how you would define a mutation.
So a mutation here, we're a bit non-committal on that. So generally it's assumed these are genetic mutations and that they confer some fitness advantage. So they make the cells proliferate faster, or they help the cells avoid the immune system or whatever. We can also include epigenetic events as well. So it's any kind of transformation that affects the cell. We distinguish between what we say in the final bullet point here, passenger mutations and drivers. So passenger mutations don't affect the phenotype. They don't affect how the cells behave, whereas driver mutations have some advantage for the cell, which allows the cells to proliferate faster, less likely to die, that kind of thing, more invasive. And driver mutations that affect the dispersal rate. We model those things separately. So I created this model. I started creating this when I was in Montpellier, and I continued it when I was in Switzerland. Various other things came along, and being able to complete this project really came down to the assistance of two very good students, Dominic Burry, who was doing his master's at ETH and is now pursuing a PhD at University of Basel, and Cecile, who was doing an internship, having finished her master's, who is now doing a PhD at EMBL in Heidelberg. The two of them helped me run many instances of these models and analyze the output, and were very instrumental in getting the paper into the form that it's in. In order to parameterize this model, what I wanted to do was base it on information from histology image analysis. Now, this is something that I know your center has more expertise in than I do. In fact, recently I wrote a behind the paper blog post for the Nature Evolution Community website. And one of the things I explained there is how I met this guy, Jacob Katter. It was just at a workshop in the Netherlands. I happened to be chatting with him on the way to the venue from the hotel. And I was explaining I got this issue that I really wanted to parameterize these models using histology data, but I didn't have the expertise. Well, it turned out that Jacob Katter has abundant expertise in image analysis using QPath and other tools, and in machine learning, but also he is a clinician with pathology training. So he really understood the pathology and how to interpret these images from the Cancer Genome Atlas as well. So he was an ideal collaborator. Oh, and he also has a background in agent-based modeling. So Jacob conducted image analysis, and what he conducted was, I think, from his viewpoint, quite simple. So there's another question in the chat. Yeah, so the question in the chat is, how do you account for the tumor promoting effect of cells in the microenvironment, such as fibroblast versus anti-tumor immune responses? So there is a fair bit of modeling that has been done by other people. Okay, people know Jacob Katter, that's great. Yeah, I'm not surprised that people know Jacob very well. So there has been a fair bit of stochastic agent-based modeling and other kinds of models looking at the effects of fibroblasts. And this is certainly something I'm interested in and something that colleagues of mine are interested in. Similarly, anti-tumor immune responses. This is another layer of complexity that can be added to these models. what I wanted to do in this particular project is create the simplest model we could, the simplest, useful, non-trivial model that has only the very, very basics in it. And then, as we discuss in the paper, there are many other things that we don't attempt to model, partly because we cannot parameterize those in general. So this is a very broad theoretical project where we try to compare many different tumor types. And things like fibroblasts and immune responses and other aspects of the microenvironment will vary between tumor types and subtypes of those tumors, so that it then becomes more complicated to draw general patterns. So what we're trying to do here is just look very generally about whether the spatial structure matters. And in order to do that, we stripped out everything else that we didn't need. But I agree, I mean, this is the ongoing work, is to add in more of these complications and to tailor these models more to particular systems. So yeah, I think what Jakob did from his point of view was much simpler than many of the other things he did, which was just delineating these regions of the tumor, these contiguous regions, and counting the number of cells. So it's a semi-automated image analysis just to count the cells in these glands. And what he found is that the number of cells in these 2D sections of the glands is generally in the order of a few hundred. And the cell density doesn't really vary much by cancer type. So this is these are slides randomly selected from the Cancer Genome Atlas across 5 patients, and then I think it's several slides per patient. And so there's there's a broad range here, but there's some consistency in the average. So this might seem quite crude. All we're doing is we're counting the number of cells in 2-dimensional patches. What we don't know, and what I understand is quite a tricky issue, is then figuring out how many cells that implies there are in 3-dimensional structures that are separated from each other. And also beyond that, working out how many of those, what proportion of those cells have unlimited proliferative potential. So we didn't address those problems explicitly because I think doing so is, these are big projects in themselves and they're beyond the scope of what we were trying to do. What I took from this was just order of magnitude estimates of how large these patches are, that they're not just a handful of cells and they're not tens of thousands of cells. And then I used, I varied parameter values by orders of magnitude, so we got a good idea of whether this parameter has a strong effect, as you'll see. Now, this idea of looking at these epithelial patches is something that seems to be catching on. So while we were conducting this project, in fact, another project was going on unbeknownst to us, which was published in 2020, looking at what they call tumor communities. So these are typically comprise 10 to 100 cells, which is similar to our estimates. Again, this is looking at 2-dimensional structures. And what this study showed, among other things, is that the cells that are within these patches are similar to one another. They're more similar to one another than to neighboring patches. So these are, in this study here, they term them tumor communities. That's equivalent to what we're looking at, these patches of cells. In the model, what population geneticists call these are DEAMs, which are local patches of cells. And in this study also, they found some evidence that these different spatial structures have some effect on patient survival and so on. So I set up the model with these parameters. We have a driver mutation rate. This is the rate at which the cells acquire beneficial mutations of either 10 to the minus 6, 10 to the minus 5, or 10 to the minus 4 per division. The central estimate there of 10 to the minus 5, this is kind of a consensus that various approaches have honed in on for typical driver mutation rates. It's an assumption that many people make, is that driver mutation rates are around 10 to the minus 5. So I went an order of magnitude either side. The driver fitness effect, I looked at a wide range there, up to 20%. And the spatial structure, the Dean carrying capacity, as I said, I considered orders of magnitude here. These are powers of 2. So all the way down to 1, then 500 or so, 2000, 8000, and also the well-mixed case, which is all the cells belong to one patch and they're all interacting with each other. The model assumes that the cells within a patch or a tumor community are all interacting and competing with each other, and they're not competing directly with the other patches. But cells are able to move between spatial locations depending on the spatial structure of the model, which is what I'm about to show you. There's an issue that depending on the Dean carrying capacity, that the size of these patches, the bigger the patch, if you set the same dispersal rate, the faster the tumor will grow. And that's not realistic. So what I did, I assumed that evolution constrains tumors to grow at certain rates because we see that human tumors grow over the map, typically over several years. So I adjusted the dispersal rates, that's a free parameter, so that all of these tumors grow at similar rates. So that is a free parameter here. What we honed in on then, in discussions with Jakob and examination of the slides and so on, is 4 canonical types of spatial structure. So the first type is blood cancers, such as AML, where we assume, and previous modelers have assumed, that a non-spatial model is a pretty good approximation of these semi-solid tumors. Now, I know that there is, in fact, spatial structure within the bone marrow and elsewhere. That means this is not wholly accurate. But what we assume is that these cells are able to mix fairly well. So the non-spatial model is a reasonable approximation. And again, this is something that many people have assumed in the past. Colorectal adenoma. This is a system that's been very well studied and is thought to grow through gland fission. So you have these glands which contain several thousand cells, and the tumor grows by these glands splitting, and the daughter glands then proliferating until they get as large as their parent, and then they split and so on. And again, this system has been recapitulated in various mathematical models in the past. The third case is solid invasive tumors, such as the slide here, which shows breast cancer. This kind of system, strangely, even though this is a dominant structure, that we see this glandular structure with cells spreading by invading neighboring tissue, there haven't been previous mathematical models based on that explicitly try to recapitulate that way of cells proliferating. So this is what we introduced, is a model in which the cells are divided into these patches, and then cells are able to move between patches at some rate. And we call this the invasive glandular model. And again, we start the model with a field of healthy tissue, and we introduce the transformed cell to the middle. So it has to proceed by invading normal tissue. So I have another question, which is, how did you learn about these spatial structures? By observation or modeling the cellular communities and so on? So we worked from the biology. So we worked from the biological literature of the pathology literature of what is known about tumor organization. And we also looked at how previous mathematical and computational modelers had modeled this system, because one of the objectives of this study was to test whether these assumptions that people have made in the past were consequential. Does it matter if someone assumed that solid tumors could be modeled using a non-spatial model? Does it matter that someone modeling colorectal carcinoma assumed that it continues to spread by gland fission rather than by invasion? Does that matter? So we looked at both the clinical and the biological literature, and also the modeling literature. And we came up with these 4 different ways of seeing things. So does this model take into account the intratumor heterogeneity of spatial structures within the same tumor? So could there be more than one of these spatial structures within the same tumor? Absolutely, yes. So again, that is another follow-up. Essentially, there are very many different directions that I could take this work, what we found is that even where you break it down into this simplest case and you say, look, we're just going to look at these 4 spatial structures, we're going to assume that the spatial structure is homogeneous, it's already rather complicated to do that thoroughly, and as you'll see the results that I come on to. So yes, it is very interesting to think about whether these spatial structures vary, and also whether the way that cells disperse changes over time as the tumor progresses. So this is something we discuss in the paper, that, for example, in colorectal tumors, what you might have is that this mode of gland fission dominates early on when you have an adenoma that really is growing into more or less empty space, into the lumen. And then later on, some of these cells acquire mutations that allow them to invade the surrounding tissue. And then once that invasion begins, you have a different kind of spatial structure emerging, which is an example of exactly what the questioner asks, which is that you have a transition from one kind of spatial structure to another. And that's something we discuss in the paper, but we don't explicitly model, partly because in order to do that, you would need further data to see when that transition occurs and why it occurs and so on. And so that becomes maybe a follow-up project that we will pursue. So finally, we looked at the case of boundary growth. And in this case, instead of the cells being organized into local patches, we have a cell that is initially located in a void, surrounded by nothingness. And this cell can then divide. And as the population grows, new cells are added to the surface. So this population of cells can only grow at its boundary. There are variants of that as well, which means that as well as growing at the boundary, internal cells can replace their neighbors as well. And we also looked at that variant. So these are known as Eden growth models and voter models in the mathematical literature. And a surprisingly large number of studies have used this kind of boundary growth model to model tumor growth. I think this is partly just because it's a very easy one to code. It's one of the easiest models to think of, that you just have this growth at the boundary. But there are also arguments that it might be appropriate for certain kind of benign tumors and perhaps certain kinds of hepatocellular carcinoma that grow as nodules with very well-defined boundaries and seem to just grow without invasion, and they just physically push the existing tissue out of the way. So these are the 4 types we settled on. As I say, this is partly from reviewing the literature, the biological and medical literature, but it's also from looking at the different ways mathematically one can set up a model. And I think it would be, okay, we have a question. No, we have a hand, yep.
Hi, Rob. Just for the sake of many people on the call who are specialists in image recognition, they're very much new to the field of mathematical modeling. Can you just explain a bit more on exactly what your input to this mathematical model would be? So I assume you have these different initial conditions and you have your different models for each of these different types. But could you just, for example, explain, you mentioned before how you have the cell density. How would you actually capture the cell density within your different mathematical models?
Yeah, so fundamentally, again, we were trying to keep things as simple as possible and as tractable as possible. So fundamentally, what we have is a grid of sites, a bit like the ceiling of the room that you're in. So we have this square grid, and each of these grid squares can be occupied by a certain number of cells. There's a carrying capacity to each of these grid squares. So cells can proliferate and turn over within these patches, but when the population of a patch exceeds its carrying capacity, when the density becomes too high, then suddenly there's a high death rate, which means that it is very likely that the next event within that patch will be the death of one cell selected at random. And what that does is it maintains the population size within each patch at around its maximum value, around this carrying capacity. And so all the cells are organized into these patches. And then there are rules defining whether cells can move between neighboring patches or whether the patch itself, the population splits in half. We also implement, when a patch splits in half, for example, we look at variant models. In some cases, patches can only divide in half if they're at the edge of the tumor. And in other cases, we say even if they're inside the tumor, regardless of where they are, they can divide in half. And in that case, they push other patches out of the way in order to do that. And similarly, when we look at the other models, like the invasive glandular and the boundary growth model, we run variants where cells are only able to divide if they're near the edge, or where they can divide throughout and replace their neighbors and so on. So we tried to look at as many variants as we could think of, given this setup of cells being divided into little square patches.
Okay, makes sense. So I guess the different kind of architectures that you're seeing here, that's accounted for the different kind of mathematical models that you're dealing with.
Exactly. Yeah. So that's the next slide. We'll show that. So, but yeah. So the non-spatial model, all we have to do is make one patch which is bigger than we need, and then all the cells are interacting within one patch. And the boundary growth model, we just shrink it down so that the carrying capacity of each patch is just one. So you only have one cell per patch, unless it divides, and then one of the two cells then dies, and so it kind of fluctuates between 1 and 2. So this is what we have then. We have these in the rows here. We have 4 different kinds of spatial structure. And we then put these into 4 different computational models. In fact, it was many more than 4, but these are the 4 main ones that I'm going to focus on. And what we see is that we get very different outcomes. So what I've picked here are just typical outcomes for these different models. The charts there with the gray lines, this shows how intratumor diversity changes over time on a log scale. So you see there are big differences in how much intratumor heterogeneity you get in terms of driver mutations. So whether you get, so in the second row there, you see that you get enormous diversity of different genotypes emerging, which have different combinations of driver mutations. In the first case, you don't get so much. What you also see, the middle column there, the prettiest column, this is showing what are called Muller plots. So what this shows is you have time on the x-axis, you have the frequency of types on the y-axis, and these areas correspond, different colored areas correspond to different driver genotypes. So different combinations of driver mutations. So initially, your initial cell has this kind of gray-brown genotype. And then as time progresses, in the second row, you see a cell acquires the mutation that corresponds to the green clone. And that green clone takes over, it achieves a selective sweep. And then within that green clone, other clones emerge, which are shown by different colors, and they spread to a greater or lesser extent as time goes on. And so in that second row, you see a growth of heterogeneity over time, greater diversity. And this is reflected in the tree, which is shown in the final column there. So the final column shows the corresponding clone tree, in which each node represents a clone at the final time point. The red node is the initial clone, and the size of each node depends on the final population size of that clone. So you can see in the first row, for example, the population is taken over by the dark green type, which becomes very large, and so the final tree has one very large node, which is not the initial type. In the final row, you see in the boundary growth model, not a lot happens in terms of driver clone evolution. And so the population is still dominated by the initial type, which is shown here in red. So we get these different modes. In the first case, we get selective sweeps on the top row. The second case, we get this kind of progressive diversification where the tree becomes more and more fan-shaped. In the third case, we get branching evolution with just sparse branching, just a couple of main branches. And in the final case, we get effectively neutral evolution. So, yep.
So does this mean, so if you look at the third row, for example, you can see in the, I forgot what you call it now, but the fourth column, so the diagram seems to be a lot less coarse. And then on the tree at the end, you have two larger blobs coming from the two smaller blobs. Does that mean there's a high proliferation rate or something? Is this what it means?
In which row, sorry?
So the third row. So for example,
Yeah.
It's a lot less coarse than the row above it.
Yeah, so the penultimate column is showing you the final spatial layout of the tumor, except in the non-spatial case is just proportions. The reason that it looks pixelated in the case of the gland fission tumor is that each of these patches there is representing 10,000 cells. Whereas in the next row, each of those patches is presenting, I think, 500 cells. So that accounts for the pixelation. You have larger glands, gives you, but within each of those glands, the color assigned to the gland in that spatial layout corresponds to the most common genotype within that gland. But within each gland, there's also a lot of diversity, and that accounts for the diversity in the tree. So what's happening in the third row is that the purple and pink types, which you see emerging, those are the ones that are most common in the final population. They emerge quite early on, and they're the ones that you see with the largest nodes in the tree. And then there's another type, which is green, which has emerged out of the purple one. And I suppose that is the next biggest node and so on. So the main message here is that the way you set up the model, the assumptions you make about the spatial structure of the mathematical model have a big effect on the tumor evolutionary mode. And the implication is that if these models are accurate, and they really do correspond to the structures of real tumors, then we would expect to see different evolutionary modes in different tumor types. So in order to compare to, we have another question. Yep. How did you get these pretty plots in the middle column? Using what data? So I created an R package for this. So if you go to my website, which is robjohnnoble.github.io, or just Google me, then you will find on my website a link to a package called ggmuller, which is an R package for plotting these things. How did you know which morphological structure is related to which mutation, and how did you get them as a progression of time? So this model is not set up to model any particular mutation with any particular driver fitness effect. So driver fitness effects depend on mutation, they also depend on context, and they seem to just also randomly vary between patients. There are other studies that have looked at very particular mutations and trying to model the exact effect of that mutation. Those models tend to be limited to one tumor type. So simultaneously, alongside my publication, another one was published from the TracerX team at the Crick. And what they did was something a little bit similar, but they focused on renal tumors. And what they did in their model was they set it up using all of their amazing TracerX data so that they could model particular mutations with particular phenotypic effects. What I did was I just assigned a distribution of fitness effects, which is just an exponential distribution, and I assigned a mean, an expected value, and then every time a driver mutation occurs, I draw from that distribution the effect of the mutation. So these driver mutations aren't particular ones. There's a link to my thank you. Yeah, this package is also on CRAN if you prefer to use CRAN, but yeah, so there is a package for drawing these things. So the data I got then was from TracerX renal, TracerX lung cancer, because these are amazing studies that have very large cohorts where they've conducted multi-region sequencing, and where they construct, as you can see in that graphical abstract on the top left, they construct these driver phylogenetic trees, which are just like the trees that I showed you in the previous slide, subject to some caveats, which we can discuss, but they're very similar. There's another study there by Yates et al. Looking at breast cancer, and a study of AML, which came from, which was analyzed by my colleagues in the Birnbinkel group, which has now been published as well. These are the initial studies I looked at. In revising the paper, I added in several more studies, which was quite nice because that corresponded to out of sample data. We'd already shared the preprint, we'd already submitted the paper, and when the reviewers said, can we have more data? I went away, got more data, and then was able to show that it was still consistent with the models, even though it was out of, it was new data that we couldn't have seen previously. So what we do then is we draw these driver phylogenetic trees for the data as well as for the model. And this is examples from the top two rows there are tracer X. And the codes here are patient codes that we used in the original publications. So the top three rows are from data, 3 solid tumor types, and the bottom is the corresponding model with our best guess parameter estimates, just for an invasive tumor, not particularly tuned to either breast, lung, or kidney, but just the general characteristics of an invasive tumor. And you can see, well, just eyeballing them, they look vaguely similar to each other. The model looks about as similar to any of the real trees as the real trees do to each other. So maybe we're in the right ballpark, but we really wanted to make this quantitative. So I developed these indices, which are pretty common sense indices. One of them is just the diversity of driver combinations. So this is just an ecological measure of diversity. Effectively, it's the number of nodes on the tree, but these are weighted by their frequency. So if you had n equally sized nodes, then, well, let's say if you had d equally sized nodes, then the value of this diversity would be d. So it's the effective number of nodes is one way of looking at it. This is the Simpson index. And the other one is the mean number of drivers per cell, which is just the average branch length on the tree, again, accounting for population sizes. And the idea was that these two measures, these two indices are quite robust to sensitivity of the sequencing and so on. And what that allows us to do is to plot all of these real tumors on two axes. One is N and one is D. We can also do some analytical work, finding, for example, there's this gray shaded out patch. It's impossible to construct any trees within that gray shaded patch. The pink trajectory at the bottom is the trajectory that would be taken by a population that experiences a sequence of selective sweeps. The curve in the middle is the maximum diversity that can be achieved in a linear tree. If you can construct linear or branching trees anywhere below that line, above that curve, you can only construct branching trees. And what we do then is we plot all these solid tumors so that the solid black points are the original data, the open circles, the ones that came in the revision. The purple squares are the data on AML. So we have solid tumors in black and leukemia in purple. And then we have the results of the model. And what we would hope is that the non-spatial model, which is shown in red on the right-hand side, corresponds to leukemia. Well, it seems to correspond fairly well. And we'd hope that the invasive glandular model, shown with the blue triangles, corresponds to the solid tumor data, where it does to some extent in that the mean number of driver mutations is similar, but the diversity we see in the model is clearly a bit higher than the diversity we see in the data. The other thing you can see here is that progressive diversification achieves higher number of driver mutations and higher diversity. The neutral models are all stuck near the bottom left-hand corner. We divide this region into areas so that we can talk about modes of evolution. What we can then do is do a lot of sensitivity analysis. So we ran many thousands of simulations on a cluster, and we looked at what happens if you vary the driver mutation rate, the driver fitness effect, and the Deem size. And we see that the model results are pretty robust to that. Some of these results for the invasive glandular model are more consistent with the data than others. You get better overlap between the colored points and the black points. Still, there seems to be a bit too much diversity in the model. However, something that TracerX found and others have found is that multi-region sequencing data fails to detect rare subclonal driver mutations. So the more biopsies you take, the more driver events you detect. And typically in this study, they only had about 6 or so biopsies per tumor, and therefore they're only getting about 75% of the mutations. And so if you allow for that, if you allow for this sensitivity and you simulate that, so when we take the data from the model, if we process the data to allow for imperfect sensitivity, then we actually find a much better agreement between the model and the data. So that's an overview of what I did in that paper. There are some other things in there as well, but that's the general idea. And the extension, why I'm interested in image analysis, is because I think a lot more can be done to understand how we can parameterize these models and make them more realistic and tailored to particular tumor types. So what I'd like to do, if I do I have time to keep going for a little bit, for another 10 minutes or so?
Yeah, please do.
Great. So the other thing that I did with this model was look at consequences of intratumor clonal diversity. So again, this was a major objective of TRACERx, was to look at whether intratumor diversity predicts shorter survival. So the idea is that if you have more diversity, you have potentially more aggressive types hiding in there somewhere, and this patient is going to have less good outcomes. And indeed, when they looked at their data in 2018, they found that this is true. If you just look at higher intratumor diversity, then you do find shorter survival. But what they also found is that if you control for stage and grade, then this effect disappears. So in other words, it's true, but it's no better than the standard, which is to look at stage and grade. And we wanted to understand why this happened and whether this is always the case. So we used the same model. And what we found a bit surprisingly is that in some cases, higher diversity can correlate with slower tumor growth. So you detect tumors that have higher diversity, and you find that in the future, they are going to grow more slowly. So that might seem a bit unexpected. Again, this was work in which a couple of master students, John and Cecile, were instrumental. The reason we found for that is quite straightforward, that higher diversity can arise because the tumor doesn't experience selective sweeps. In the opposite case, as shown here, you have a tumor that has a succession of these selective sweeps. It acquires a lot of driver mutations. And in the top row there, what you see is each color is a different clone as before. In the bottom row, what you see is the color getting more and more red corresponds to higher and higher division rate. And because you get these selective sweeps, the selective sweeps eliminate the diversity. The same way that Omicron swept through, it wiped out all the diversity that there was in coronavirus variants previously and replaced it with these Omicron variants. So it wiped out a lot of diversity, but at the same time, this is a variant that was spreading much faster. We see a similar effect here. So you can have low diversity and high division rate. And similarly, in the opposite case, if there are no selective sweeps, you can get this slow progressive diversification happening, and the division rate doesn't change much, and these patients, the tumor will grow more slowly. So this is the invasive glandular variant of the model. So that's just a summary of what I said. In TRACERx, they found that if they accounted for genomic instability, so the fact that some of these tumors were more unstable than others, then they found that that was also a predictor of survival. And if they combined both intratumor heterogeneity and genomic instability together, they found that the slowest progressing patients were the ones that had low instability and low intratumor heterogeneity, shown in black here. So what we did in the model was we saw whether we could also forecast progression-free survival. So we grew tumors, simulated tumors, then these tumors could acquire not only driver mutations, but also rarely resistance mutations, which had no phenotypic effect. And then we introduced the treatment, and when that happened, we eliminated all or nearly all of the cells. And if the tumor grew back, we measured how long it took to grow back. And we saw whether the intratumor heterogeneity just before treatment predicted how rapidly the tumor grew back. And what we found is that clonal diversity by itself was not a particularly good predictor, but if we combined it with intratumor heterogeneity, then we got this similarly, qualitatively similar results to TRACERx. So top row is our model and bottom row is TRACERx, and they're qualitatively consistent. But what we again found is that this is largely because the intratumor diversity is is a consequence of the instability. It becomes a proxy for the instability. So we were able to look at this in a Cox proportional hazards model, and to some extent explain what might have been going on in their studies. I'm going to talk very, very briefly about a final spin-off project. So this project came out because one of the reviewers of the first paper I discussed said, why are you just looking at these indices N and D? Shouldn't you also look at tree balance? That is, not just like how long the branches are and how many nodes there are, but also whether the tree is imbalanced. And what we found was that, in fact, this is a very interesting question, but that all the methods that have been produced to do this over the years were insufficient for what we wanted to do. So the basic idea is that a tree can be balanced or imbalanced. The one in the top left here is imbalanced, and the other ones are considered balanced. It's quite a straightforward notion. And there are various indices to look at this. So the most popular is called Sakins index, which just counts the depth of the nodes and adds them all up. So the nodes here all have numbers in them corresponding to their depth, how far you have to count back to get to the root, which is shown in red. You add all these up. Now, obviously, this is a bit of a problem because as you get more and more nodes in the tree, even if your tree is balanced, this index is going to increase. So there are various ways of normalizing it. But a big problem is that these current indices are only defined on cladograms. And what we have in tumors are like this, and they look nothing like the cladograms that people interested in species evolution are used to looking at. So with a very talented master student, Jean Lemont in Basel, I looked at developing a new tree balance index that was much more generally applicable. So we started off with the idea that a linear tree should have balance 0 because it's considered the extreme of an imbalanced tree. A tree in which every branching event gives rise to descendant nodes of equal size, such as shown in the middle here, has perfect balance. So the maximum value J equals 1. So what you see here is that nodes can have different sizes corresponding to the population size of the clones that correspond to them, as we saw in the tracer X trees and the ones from our model. And then we generated some other axioms that guarantee insensitivity to small changes, which we wanted this robustness so that we're not too affected by sensitivity of sequencing methods. And my student Vess is continuing this work. So what we ended up with was this index here. And what this does is it just takes the Shannon entropy. So I understand there are some computer scientists in the audience and perhaps some people with background in physics, so they might be familiar with Shannon entropy, which is just P times log of P, where P is a proportion. And in this case, Pij is the size of a branch of a tree. So I'm not going to go into this in too much detail, but what we find is this index, which might look complicated, it's actually quite a straightforward notion that we're taking the sizes of these branches of the subtrees and we sum them up and then we normalize them, which is this factor at the beginning. And what we get then is a much more general tree balance index. And it simplifies on cladograms because in this case, instead of subtree sizes, we have numbers of leaves per tree. And the nice thing about this, you can rank trees on a certain number of leaves. You can compare trees fairly with different numbers of leaves. It overcomes a lot of limitations of previous tree balance indices. And it also, we were able to show that it generalizes Sakins index, this other popular index, because with about a page worth of working, you can show that this index, in the case of cladograms in which all nodes have the same out degree, is inversely proportional to Sakins index. So it can be seen as a generalization of Sakins index. It allows you to compare different models and so on. And it correlates with previous indices. And it's robust. So what we then did, we were able to use this new index also to compare our clone trees. And this is all just to say, I mean, apart from the interest that computer scientists might have in this notion, is that what we now have is a set of indices for comparing these modes of evolution. We can compare these trees in different dimensions, and we can get a better idea for comparing models and data. So I'll just wrap up and say what we find is the main result of the first study is that differences in spatial structure suffice to generate a spectrum of tumor evolutionary modes, that it's consistent with clinical data. So if the message here for computational modelers is that the way you set up your model really matters. And the message for clinicians is that the spatial structure of the tumor is likely to have a powerful effect on clinical outcomes. And we really need to pay a lot more attention to what we can see in these pathology slides. And that's why I'm interested in the power of image analysis to uncover what information there is in these slides and other data. We find that these prognostic biomarkers, like the degree of intertumor heterogeneity, they could be useful, but they're quite complicated to interpret in some cases. You have to be a bit careful. And we've got these new evolutionary indices as well. I have a question here, which is, it'd be interesting to consider virus-associated tumors where there are fewer driver mutations. So the viral genes correspond to driver mutations, such as Epstein-Barr virus. Yes. Okay, and very rapidly growing pediatric tumors as well. Yes, I mean, what we went for here were kind of canonical tumor types. And I agree that there are many other types that we could look at. There are also various other precancerous conditions that we can look at as well, which we haven't done yet. But yes, it's a very nice suggestion. Thank you. So I will just wrap it up by thanking the people who participated. So my former bosses, Nico and Mike, my students who are all in blue there, and my collaborators, Jakob and Yannick, shown in red. Yeah, there's more information on my website and Twitter and so on. The Arizona Cancer Evolution Center provided a lot of funding to support this, and there are 3 papers there where you can read more. Thank you.
Great, thanks, Rob. Brilliant presentation. Really interesting. So if you don't mind, maybe just a few of us have a few more questions. I'm not sure how much time you have. But maybe
sure, yeah, I'm happy to wait.
Yeah, brilliant. So I know we have another comment or question online from Lawrence, and it says, can spatial structures relate in any way to metastatic potential and to resistance to therapy?
So metastatic potential is a very big question. It is very difficult to find anything that correlates with metastatic potential. It is obviously the million-dollar question, right? Because everyone wants to know what correlates with metastatic potential. It is not something I investigated in this case. You can imagine that spatial structure might influence this, and there have been some studies that have looked at that. They're making certain assumptions, but one of the issues is that we just don't have that much data on why some metastatic cells are successful and others aren't. But for example, the group that I was in Basel, they collaborated on a project that showed that clusters of circulating tumor cells are more likely to establish metastases than single circulating tumor cells. So that, and there are other studies showing that the initial community of the metastatic cell matters. This is like the seed versus soil debate. That is not necessarily a genetic mutation. It is the circumstances that the cell finds itself in, its ecological niche, and if it's traveled along with other cells, then that can be beneficial to it. This is not something I've explicitly modeled, but yes, there's certainly potential there. And also the structure of the tissue that it arrives in. To resistance to therapy, yeah, there's a whole other thread of my research is looking at how to suppress resistance to cancer therapies. One of the big ideas that we pursue there is something called tumor containment, also known as adaptive therapy. So the idea is that if you have a tumor that has pre-existing resistance, then rather than trying to eliminate the tumor and getting relapse due to these rare resistant cells then regrowing and repopulating the tumor, you might get better outcomes instead by shrinking the tumor to some acceptable tumor burden, which is something that has to be discussed between patient and physician, and then maintaining the tumor at that size. And then what you get is competition between treatment sensitive and resistant cells. You get ecological competition, and then the idea is that you can contain the tumor and you can keep those resistant cells competing with the sensitive cells and not growing out. And this is something that has been looked at for a few years now. And we published another paper on that last year, which again, you can find the link on my website, where we look very generally at mathematical models of that idea. And again, it does come down to spatial structure. And so we're conducting further experiments and models to understand how that relates to what went on in this project.
Great. Can you maybe just say a few words as well about how exactly you would plan to incorporate image analysis into your pipelines? Would it be to have extra prior knowledge into your parametric models?
Yeah, I'm also interested in looking within tumor types. So we know that within certain tumor types, the spatial structure can be prognostic, like the extent of invasion or the nature of invasion. This is again something I discussed very briefly in the paper, is studies that have shown that. And this has been shown particularly in colorectal cancer, I believe. And so what would be very nice is to be able to look at that spatial information that we can get from image analysis, and to couple that with models that are more tailored to that particular tumor type, and then say, do the clinical outcomes vary depending on the spatial structure, and can we recapitulate that in the model, or are we missing something? Is that in itself enough to explain the variation we find? So I have a colleague at City in the computer science department there called Carlos Reyes, who works on also image analysis. And so this is something we're discussing, is pursuing that kind of direction. And this is something I'm also discussing with the Kata.
Yeah, because you spoke about having these different models for different spatial organizations that you see. And you spoke about glandular organization as well. So I guess if you could localize the glands using image analysis, then that could be really interesting, couldn't it?
Exactly, yeah. So that's what Jakob did so far, is just to count the number of cells per gland. But we'd love to be able to do this in 3 dimensions. And we'd also love to have something less of a static viewpoint. It would be great to have more information about how cells are moving through the tissue. Do they invade by themselves, or in many cases, the studies that I've read find that in fact it's groups of cells that are moving together, and they make promontories and they invade that way. And that's something we haven't explicitly modeled as such. But that information about what's going on in 3 dimensions is, yeah, I think there's a lot more that we can extract from images that we haven't done yet.
Yeah, great. So I think one more question in the chat. So Lawrence said, could you use in vitro organoid culture systems to test and refine your model?
Yeah, I think what's being done with tumor organoids is fantastic. I don't currently have any collaborators that do that. I have colleagues that work with tumor spheroids, so that is something I've modeled, and something that my students are continuing to model is tumor spheroids, which are much simpler. Yeah, I'd be fascinated by modeling organoids. Basically, if anyone has suggestions and they'd like to approach me, then by all means email me, and I'd love to discuss more about other systems and other kinds of analysis that we can incorporate into these kind of models.
Brilliant. So I think maybe one question just to tie it all together. So all of this work that you've done looking at the evolution of cancer, can you maybe just say a few words to describe how it can have an impact on the patient at the end?
Yeah, so what we hope is the ultimate idea is to have a prognostic model so that you get some standard clinical information, and that allows you to forecast what is likely to happen in that patient. And really, I'd like that to be mechanistic. So at the moment, a lot of these models are just correlational. We say, if we see this certain property, if we see this mutation or whatever, then we assign it to a subtype. But we don't have a mechanistic explanation for why that should be. And what I'd like to have is something that relies on these biomarkers, like the extent of heterogeneity, the speed of turnover within the tumor, that kind of thing, and the spatial structure. And then we feed that into a mechanistic model. And that allows us to predict things in the same way, more like weather forecasting, where instead of just saying red sky at night is going to, it's not going to rain tomorrow, We say, no, we put this into a system of equations and it spits out some probabilities. And we say, oh, there's a 10% chance of rain or tomorrow. Not because we've done it on rules of thumb or whatever, but because we have an actual effective mechanistic model. So that's one goal. The other one is to design more effective, evolutionarily informed treatment strategies. And that's a thread of my research that I haven't talked about, but it's another major goal of ours, is looking at how we can exploit these interactions within tumors. And potentially that means we can give a lot less drug. That's one of the big benefits. And so you have fewer side effects, better quality of life. Instead of trying to blitz patients with the maximum tolerated dose, you use lower doses in a more intelligent way to steer the growth and the evolution of the tumor. And maybe the dream there could be to turn cancer into a chronic disease rather than something that's deadly.
Great, thank you very much. Anybody here got any questions? So we also have 10 more people in the room. So of course you can't see those people, but there's 10 more people joining me here in the room as well. So yeah, I don't think there's any more questions. So I think with that, we can wrap things up. Thanks again, Rob. Really exciting presentation, and have a great rest of your day.
Well, thank you. Thank you for hosting me. It's been fun. Thanks. And thanks for the great questions. They're very good ones.
Great. Cheers, Dan.
Thanks, Rob.
See you.